Now we're recording. All right. Everybody see scientific literacy on the screen? Perfect. All right. So let's talk a little bit about what scientific literacy is. Essentially, it's being able to understand, read, and participate in scientific conversations. And for the general population, this kind of means being able to make personal decisions. Um, I know that as soon as I became a biology major and was kind of fascinated with this, every medical appointment that my family goes to, I get a call or a text saying, "Hey, what does this mean?" I don't know if that's similar in your family. There's just some of that biology that you pick up on, but like when they first started asking me, I was no way an expert. And still, when they ask about different conditions, I'm still not an expert in different diseases, but you know, I'm able to take some of the information that they have and put it into context to kind of talk about it. And um, it gets a little bit more complicated when cultural beliefs, different political beliefs, things get intertwined in some of that. And we've noticed that over the last couple of years. So it's just kind of the ability to read scientific literature, understand, and apply those concepts. So there are three common elements to scientific literacy: knowledge of scientific content, understanding science as a way of knowing, and understanding and conducting scientific inquiry. All right. Um, Essentially, by being able to, I'm going to move this bar over here. I don't know. Oh, sorry. Your faces are kind of in the field of the writing, so I have to make sure I write. All right. So essentially, can you pose questions? Are these claims credible? So when you hear a claim, whether it's on social media or on the news, you know, is it credible? Is there any evidence to support the claim? What reasoning was used to justify the claim? Is the evidence reliable? Was it gained in a rigorous manner? Is the claim being manipulated or misrepresented? All right, so let's look at an example. One of the most prominent examples that I have noticed over the last couple of years was that there is a misinformation campaign, most notably on social media, um, about how the COVID vaccine alters your DNA. I don't know how many of you have heard that, but that was one of the reasons people were not taking the COVID vaccine, because they heard it altered your DNA. All right. So as a scientist, you have to like kind of look at it. Does that make sense? We know it's an mRNA vaccine. Okay. So, you have to think back. Do you refresh your biology? You remember that DNA gets transcribed into mRNA, which then gets transcribed into protein. Okay. So if you're putting mRNA in, the first instinct would be, no, that doesn't make sense. And if you want to go into more of an upper level biology um, look at it, you can look at how it's transported. For mRNA is transported from the nucleus, and is it a two-way kind of street? And most of the modeling shows you the mRNA going from the nucleus to the cytoplasm. All right. All right. Can mRNA get into the nucleus? I guess it's possible. I mean, you don't, even though I know that central dogma of DNA goes to mRNA goes to protein, I always know that there are different circumstances and different things that are going on. And when you deal with viruses, some of that gets thrown out the window. So take a deeper look. When I look at the mRNA vaccine, so in a normal cell, again, DNA to mRNA to protein, in the vaccine, particularly the vaccine that we're looking at, it is a in the vaccine, it had about a 4 kilobyte or kilobase pair sequence that was used for the vaccine. The whole virus itself is 27 to 30 kilobytes. All right, so the vaccine is encapsulated mRNA, gets into the cell, it makes proteins. Those proteins then trigger the immune system. All right, that makes sense. So why are they, I wasn't sure, why are they looking at essentially it altering your DNA? It just didn't make sense. So go to the original paper. The original paper that this came out of, because even misinformation oftentimes, and you see this with different product developments, has some resemblance of plausibility and truth, right? There's something in there that they have to pull from. And like I get asked all the time, like some of those cloths with the silver in that, how effective that is in preventing bacteria and antibacterial properties. And it's true. Like you put silver nitrate on, that they have ointment that they put on eyes when babies are born to prevent infection in the eye. So silver is a natural antibacterial. All right. How effective is it in a washcloth? I don't know. That would be a separate issue. So there's some plausibility about silver being, or silver nitrate being kind of an antibacterial, but how does that function? So I looked at the paper for kind of the original paper that caused such a stir. And in the abstract, they talk about, so they use these liver cells, and HUH7 are essentially liver as a liver cell line. And if you don't know what a cell line is, these are cells that kind of grow indefinitely. Most of the time they have properties similar to cancer cells in that they can keep dividing, keep dividing, keep dividing. And so you can keep growing them out. They don't have what's called cellular senescence. So they don't have a natural aging or dying process like you do see in kind of most cells of the body. All right, so cell lines allow you to look at a particular experiment at the cellular level, and see how whatever your treatment does affects the cell, right? They're not in the body. These are experimental cultures. So essentially, they looked at this BNT 16 or 162b2, which is that gene sequence that was in the vaccine. All right. So they were looking for that particular sequence that's unique to COVID. And they were looking at where it came up on the cell line. And what they found is they found an increased nucleus distribution of the line 1, indicating that those products from that mRNA vaccine were found in the nucleus. Okay. So that was their observation in the lab. They had no evidence that it altered DNA, but they had evidence that it was there. So it was present. Okay. And that's what caused kind of this stir. Well, then, so you had the tweet, it's altering your DNA, it's manipulating your DNA, it's getting into your DNA. That causes kind of the Associated Press and kind of more of the non-scientific community to get involved. They start talking to scientists. The paper itself is getting a lot of press, but there was no evidence that the DNA fragments from the vaccine were getting actually incorporated into the DNA. There's also no translation for a cell line into the body. It behaves mostly like a human cell, but they don't know for sure. All right, so then you get retractions, the claims come back, more retractions, or not retractions, but more comments on it, more looking at it. Other scientists were looking at, is this possible? Doing kind of the same thing that I was doing, kind of looking more in detail. It was such a big campaign that they had to put it on to the NIH CDC website saying that this is not happening. We had to have a lot of fact checks during this time. All right. So what are your thoughts? Let's open it up a little bit. What do you think supported the claim? What do you think refuted the claim?
I think probably a lack of scientific literacy led to a lot of assumptions.
Mm,
Um, and I think when you have these sensationalized headlines, a lot of people jump on it because it's exciting and it's new and the general population probably has a harder time distinguishing between this.
yeah. Yeah. I mean, did any of you experience what, like, you saw a misinformation campaign like that and it didn't make sense? Or, I mean, do you take the time to look into some of that?
I mean, I definitely saw some of that misinformation and kind of was able to set it aside. And I, granted, was a little more informed on the situation. I feel like I sort of was at the level that you just explained, and it's like, well, how is it being incorporated into the DNA, right? Like, there's no system for that, you know?
Right. And in one of the big things for me, like, it was in general, human cells don't have, like, I had to go look, they don't have a reverse transcriptase like HIV does, right? HIV is an mRNA virus that's able to reverse transcribe into and then incorporate into the genome. Um, and so I had to look, and there are some cases of some endogenous reverse transcriptase in human cells. Um, and then, I had to go back and look and say, well, wait a minute, we're getting a $4,000 or $4,000 base pair fragment of that whole virus. So if that's happening, um, for me in my head, wouldn't it be more likely that the whole virus in a natural infection would cause changes? Because some of that endogenous reverse transcriptase is actually like viral remnants from evolutionary biology. Either way, you can do that whole, like, well, it's possible, this is possible, that kind of stuff. But then in the end, are you seeing anything? Right. And that's kind of that bigger deal. Like, I have some family members that I have to kind of explain stuff to, and some of the stuff they don't want to hear because they're, guided one way or the other. And it's just a matter of coming to a term, and you might not get an absolute answer, and it might change as we get more scientific information, right? And that's the beauty of science, is that as we get more information, we can adjust what we were thinking before. So if I see more and more papers come out saying, yeah, it inserts into chromosome 12, it causes no damage, or it inserts in chromosome 12 and we see, um, an effect on these populations for whatever reason, okay, I'll change my mind. All right, that's happening. Does it, and then you have to start, does it outweigh the benefits or effects of the vaccine itself? Right. And it's never as clear cut as you want it. A lot of us want a yes or a no. And what I've found in research is I really get a full yes and no. So, all right. Any other thoughts on that topic or misinformation campaigns? And no? Okay. All right. I kind of looked into a little, or talked about this a little bit in a different way, but we can have bias in a scientific study, and there are different degrees of bias. So bias is any trend or deviation from the truth in data collection, data analysis, interpretation, or publication to lead to false conclusions. This can be unintentional or intentional. Now, this can happen at any phase of your research. So if you start a project wanting to prove a specific result, right? You're going to look for ways to prove that specific result. Maybe not do the best experimental design, right? To give you more of the truth of what's going on. And so um it can happen, the bias kind of happens. Sometimes it's pressure for a grad student or a researcher to get a certain result, and they want to publish because their doctorate degree depends on it. Like, this is when they can finish, or this is the grant that they're writing, depends on this information. And we have to be very, very, very careful that we're not being biased during a study. So we want to avoid bias. All right, so let's give an example of this is volunteer bias. It's a type of sampling bias. We want to assess the average number of heat-sensitive C-reactive protein concentration in healthy populations of Croatia. All right, so how should the samples be gathered? Essentially, what you should do is recruit healthy individuals from the general population during their regular annual health checkup. Now, in the example of the study that I looked at, it had bias sampling. It recruits only volunteers on blood donors, right? Because sometimes it's hard to get volunteers. So who is your population of volunteers? And if you have a bunch of people coming in that are blood donors because they're volunteering for this, and you can get samples easily, is that the best way to sample the general population? So blood donors are generally healthy individuals who are not suffering from any condition or illness. So that might cause changes in this heat sensitive or high sensitive C-reactive protein. So by recruiting only healthy blood donors, we might conclude that this high sensitive C-reactive protein is much lower than it really is. This is kind of a type of sampling bias. So when you're looking at papers, are they recruiting the best sample? Is this the best population to look at? And this is part of that critical reading that we need to kind of look at. And so be aware of bias in some of the samples. Here's another example. This is misclassification bias. It's another type of sampling bias. Assess the number of patients with Morgellons disease. And I put in what Morgellons disease. Essentially, it's misunderstood. It's relatively uncommon. It doesn't always have exact signs and symptoms that you can objectively measure. It's not easily classified, right? And so it's a controversial condition. It involves skin lesions. People tend to feel like there are bugs or different things crawling under their skin, or it feels like there's strings under their skin. Symptoms generally include skin rashes, a sense of intense itching, crawling under the skin, and fatigue. And, the sense of itching and crawling and the fatigue are subjective, right? They're based off of the patient's understanding of how they feel. All right. So how should you gather samples in this case? To ensure the sample is being representative of a population, sampling should be based on objective signs that every subject needs to have in order to be included in the study. However, if you're based off of who got diagnosed with that in the hospital or in a clinic, and you're just using their patient files, they're coded as having Morgellons, okay? You don't know what all went into them being officially diagnosed with it. So there's no gold standard for that diagnosis of the disease. Some might not be easily detectable, which means that you're going to lose a lot of that population if it's not diagnosed. And some doctors might not, because it's a controversial disease, they may be hesitant to diagnose that, even though that they think that that might be what's going on. All right, let's look at a different example. Selection bias. You want to assess general population health by offering incentives with health insurance for participating in an online health program. And I don't know how many of you guys work with your health insurance, but a lot of times they offer, you can get a discount on your copay or different things like that if you participate in this online program. To ensure that sampling is representative of population, sampling should be random. Every subject needs to have an equal probability of being included in the study. Well, here's during that recruitment, which patients are more likely to participate in that? I know if I had a kind of a general health issue that I didn't want to deal with, so uh, say I was a smoker, right? I probably wouldn't want to participate in this because I don't want to quit and I don't want to talk to somebody about quitting in order to get that discount. Is that discount worth it to me? Right. And so there can be lots of different reasons. They might not have time, whatever it is. So there are going to be populations that don't participate in this study. And if their health is generally has some other types of conditions, that's not going to be representative of that population. Does that make sense? Okay. Let's look at a different type of bias here. We have survivor bias. All right. So in this study, to assess the association of altered human kallikrein 6 expression with a 10-year incidence of Alzheimer's disease. All right. Here's where the sampling is biased. Subjects who died before the study end might be missed from the endpoint of the study. When you're studying a terminal illness and you're studying it for a long term, you're going to miss people at that endpoint of your study. It usually occurs in cross-sectional studies, the survivor bias, when you analyze data from a population at a single point in time, right? Because you're not going to get individuals that, not all individuals are going to have the same survival rate. All right. What do you think you would do? How would you try to eliminate that bias? Just brainstorm.
I'm really unsure.
Okay. All right. So let's look back at the aim of the study. We want to look at the expression with a 10-year incidence of Alzheimer's. So essentially, you want to make sure you get patients that have had Alzheimer's for 10 years. You could do case crossover designs and compare that individual to themselves. So what essentially, if they get an early diagnosis, can you look at the expression of that human kallikrein expression at initial diagnosis versus 5 years versus 10 years or yearly to kind of monitor that? And then that will, you can parallel that with individuals as they're going on and off treatments. And so you kind of study those individuals for 10 years, but you have to be careful of how you interpret that data. And an inception cohort is a type of cohort that involves enrolling subjects shortly after the start of exposure of interest. And then this exposure could be the onset of symptoms, a diagnosis, or detection of a significant pathological event. So it gets a little bit more complicated when you have a population that might die off before you're finished. All right.
Do you have to dismiss the data collected from people if they die off before the 10 years?
It depends on how you're reporting it, right? So what you could look at maybe doing, comparing the 5-year mark where you had more populations and what that looked like versus the 10-year mark. And note the significant decline in the population sample size. Because by the 10-year mark, depending on what your initial population was, you might not have enough to even do statistical analysis on that 10-year mark.
So if it was like a pharmaceutical study
Mm-hmm.
on the effects on Alzheimer's,
Yeah.
um, you would definitely want to include that data.
Yes.
Okay.
But you have to be careful of how you present it. Make sure you present it accurately and truthfully, and like you're honest and upfront about the population dying off, that you don't might not have a significant population to do statistical analysis. And so the results may be inconclusive, um, but you can still publish those results.
Okay.
Bias in data analysis. So when you're in grad school, you're working on a research project, you have kind of a hypothesis in mind, you have what you kind of want it to look like. Where can bias be introduced during analysis? You could report non-existing data. So this is, you could completely make it up. Data fabrication. You're adding numbers to your data set that aren't really there. Okay. That's kind of like a blatant, don't do that. Eliminating data which do not support your hypothesis. So if you have some outliers, like say you're doing statistical analysis and your p-value is where it needs to be, or is just, it's not where it needs to be, but then you have these outliers that are kind of dragging it. And there's probably like 2 or 3 data points out of 200, but that was enough to push it where the p-value didn't line up to be significant. You can't eliminate the outliers just because it doesn't fit into your data set, right? So you have to acknowledge the outliers. Using inappropriate statistical tests to test your data. I am not the best at statistics and picking statistical tests. But like when you are not using the right test for what you're looking at, it's not going to be helpful. It may show something that isn't really there. Performing multiple tests. So say you're not getting your answer and you just keep doing that test over and over and over. And out of, I do it 10 times and one of the 10, I actually got the answer that I wanted. That's not what I want to do. You have to like use all of your data. And so you're not trying to find something that's statistically significant. All right. Bias in data interpretation. So discussing, there are different examples of bias in data interpretation, discussing observed differences in association, even if they're not statistically significant. So say you're doing an experiment and we're looking at that protein in Alzheimer's disease, and you notice there's an increase, but it's within normal range. But because your brain is already like focused on that one protein, you keep talking about the impact that that increase has, even though it's not statistically significant. I would say that's more unintentional bias because you really want to prove your results. But you may be discussing differences which are statistically significant but are not clinically meaningful. So like when we were looking at that DNA is observed, those DNA fragments are observed in the nucleus of those liver cells for the COVID vaccine. Yeah, you're seeing it and you're seeing enough to say that it's present, but it has no clinical impact. It's not being incorporated into the DNA, nothing is happening with it. But you keep talking about how the DNA is present, the COVID DNA is present in the nucleus. Okay. Drawing conclusions about causality, even if the study was not designed as an experiment. I don't know how many you've gone and talked about causality, correlation. Just because something is happening at the same time doesn't mean that it's a causal relationship or it's actually correlated and has an actual effect. Drawing conclusions about values outside the range of the observed. So like you're looking at different values that are higher than normal or are extremely lower than normal. Um, and you're hypothesizing without any evidence that this is due to that patient being on this drug, right? They have a low level of the protein because of this drug. You don't know that for sure, right? And unless you're going to um, eliminate and provide evidence for that, you probably shouldn't draw a conclusion of why it was so low. There are possibilities, and sometimes people speculate in some of the discussions. But usually they speculate with evidence backing it up, right? So if I'm going to speculate that the protein was low because of the drugs that they were in my conclusion, and I could say it's a possibility that the drugs influence the level of protein as seen in this paper and this paper, right? I'm backing it up. But me just speculating isn't providing any help. Overgeneralization of the study conclusions in the entire general population. If, say I am looking at, um, let's see, I'm looking at adolescents from 12 to 16 that were, or that are females, right? I mean, if I put that to the general population and saying that we also like this could be a problem in this population or this population, if I don't have any evidence backing it up, I shouldn't draw conclusions about different populations. All right, and then just statistically, there are type 1 and type 2 errors. Type 1 error is the expected effect is found significant when there is actually none. And type 2 is the expected effect is found not significant when there's actually an actual present. It is actually present. And then this would be more into whatever, depending on your p-values, what programs you're running, what statistical analysis, just to try to make sure that you don't have some of these types of errors. All right. Publication bias. There is some publication bias. Scientific journals are more likely to accept publications which report some positive than a study with negative findings. So I found that this gene is necessary for development from the fetus of, 3 to 5 weeks. Rather than publishing an article saying that this gene has no effect on fetal development. It's useful information because it may help other scientists not waste their time with some of those experiments. If you're finding articles that they're publishing negative results, use that information because that's valuable information. You could probably not waste your time, or you can look at their experiment and say, you know what? I wonder if that would be better if they would have used this population instead of this, or if they would have used a different type of testing, and maybe I could have found that differently. So you can learn from that, but most journals, they tend to lean towards publishing reports that have some type of positive effect or positive result. All right. Personal bias. Personal bias, any learned beliefs, opinions, attitudes that people are unaware of, and reinforce stereotypes. This personal bias are unintentional, automatic, and inbuilt into leading to incorrect judgments. And this, you may not be aware of your own personal bias. If there are different things that you tend to, how do I say this? Like sometimes there are cognitive dissonance. Do you know what cognitive dissonance? You can believe in, so it's kind of a separation of, I'm trying to think of, I've been using the vaccine as kind of the example, but I believe in scientists and I believe in medicine, I believe in doctors, but the COVID vaccine is wrong and whatever the doctors are saying about it is wrong, right? There's a dissonance between there. Yes, you tend to go with all of that, but when it comes to this one thing, for whatever reason, whatever that vaccine hesitancy comes from, there's a cognitive dissonance, right? Because you can't believe what other people are saying because it's kind of just built in. Does that make sense? And we all have some of this to some degree. And just trying to be aware of it. Tend not to, I tend to be all science all the time. And so anything that's not science, I tend to like just blow off, even though it's possibly true and very valuable and helpful, right? Um, so keep in mind that you may have some personal bias. All right. So how do we trust information, right? How do you know if a source is reliable? And this is where we kind of get into different types of sources of science. Um, there are kind of two ways that you can get science information: the science for professionals, the science for the entire society. The science for the entire society tends to be newspapers, social media, different social networks, different organizations, the WHO, the CDC. They try to get information to the public, and they try to do it in a way that doesn't use a lot of scientific jargon, right? Because I will tell you that even when I read certain scientific papers, if I'm not familiar with that area, it all starts to blend together. And I can start a sentence, and by the end of the sentence, I'm like, wait, what did I just read? Like I know each word individually, but somehow strung together, it just doesn't make sense. And so if you imagine that, if you haven't had any scientific background, trying to read a science paper, it's going to be difficult. And the more you read, the more it helps. But I read science journals all the time. And if I'm in a field that I'm not into, or if I'm reading something about astrophysics, I there's something about it I just don't get, and I have to like it takes me a while to understand, and I still don't feel like I have a good understanding by the time I finish the article. All right, so let's talk about the difference between scholarly and popular. A scholarly source is typically written to share original research. It is written to synthesize the state of research in a given field. Popular sources are written to be informative or entertaining. So I'm giving the example of when the genome work came out in the 90s, there was work obviously published in the scholarly sources, but then we were informing the public as well in different sources like Newsweek. And the two articles are very, very different. The authors of the scholarly tend to have PhDs associated with a university or institute, whereas in Newsweek, the authors typically not a scientist. Sometimes there are. There are scientists on staff at different news organizations that help to write that, but they're trying to write it in a way that it's more less jargon and more friendly to the public. So who's the audience for? Is it for other scientists, or is it for the general population? Is the language complex with a lot of jargon, or is it more simplified? Where is it the publishing? Does it come from a book? Does it come from a newspaper? Different things like that. And the references. In the Nature article, you'll find a bunch of different references of different articles that they use to base the research on, and that they're using to discuss how it impacts other fields of research. In the Newsweek, you may have a link to the article online, so it might link to the Nature article, but you're not going to have a lot of references. It is the journalist opinion and kind of how their take on the human genome and human genome work, not necessarily based on facts. Does that make sense? You guys are so quiet. All right. So let's dig deeper into the scholarly articles. Scholarly articles can come from a primary source or a secondary source. Primary source, in general, is the researchers that were doing the work. They have written the work up, published a paper, and that is the primary material. Secondary sources tend to take commentaries or tend to look at, be more like review articles, where they're looking at other people's work and they're comparing, multiple different primary sources. And they can be just as valuable, because that actually, and I find that especially early on in a project, it helps to look at some of those review articles to kind of give you an overview of the field, and some of the work that's being done. Primary sources are published first, and then secondary sources are usually later when they've got a multiple of sources to come from. Formats, primary sources, they can be journal articles, they can be dissertations, interviews, lab notebooks, conference papers, things like that. And then the secondary sources are typically review articles and in magazines or books. All right. Oops, I went back. All right. Example of a scientist. Example includes a scientist studying genetically modified foods. The article in the scholarly journal is reporting the research and the methodology. So if I'm looking at genetically modified foods, what genes was I looking at? How did I go about it? What were my protocols? All of that. What were my results? How did my results look? And then if there was a secondary source, it would be an article analyzing my work compared to other work. All right. Peer review. Peer review is kind of the gold standard. And I should have probably moved this slide a little bit later, and I will. Actually, we'll go back. How do you know that an article is something that's reliable? One of the things that people do is use the CRAP test, right? They look at currency, what's the timeliness of the information, the relevance, is it important to your needs, the authority, is it researchers, or is it somebody else in the organization, accuracy, is the content accurate, and the purpose. What is the purpose of it? Is it to inform? Is it some type of propaganda? There are lots of different things. So keep that in mind. And then we're going to go back to peer review. Most scientific articles that when I'm talking about it are peer-reviewed articles. And what is peer review or refereeing, depending on which database you're looking at? Essentially, if I'm going to publish an article, so I do a study and I'm going to publish that article. So I write it up, I've got everything, I think it's accurate, all that. Then it goes to, I submit it to a journal, and then they submit it. They ask, anywhere from 5 to 10 other scientists in that field to take a look at the article. Does this look right? How are the experiments done? Is it appropriate for this journal? Is it relevant to the work that we're doing of the audience? Is the quality, free, well written, and is it original, right? These scientists that work in that field, that read these articles all the time, are then going to analyze my work to see if I did what I'm supposed to do. Like, they're looking for bias. They're looking for errors in my work. And if they accept and say, yes, everything looks above board, it looks like they were doing what they were supposed to do, the data looks right, whatever it is, then they'll go ahead and publish. So depending on what journal that you're publishing to, most scientific journals undergo peer review. Some do not. Okay. In which case, then it's going to be on you to decide the quality of that work. So the peer review kind of takes away some of that pressure to decide if it's quality work or not. All right, and so that I'm not talking all the time and I can go get coffee, we're going to watch this video.
Striving for A's? Information to support your essay.
Oh. Okay.
It can
Hit the
be
wrong
found in many different places. Consider the source. Do you have the best sources for your essay? Articles published in peer-reviewed journals are the most in-depth, authoritative sources. Using them will please your professor and will improve your chances of getting a good grade. Peer review is a quality control process used by journals in order to ensure that the articles they publish represent the best scholarship currently available. Peer-reviewed scholarly journals contain articles written by and addressed to experts in a specific discipline. They contain original research and communicate this information to other researchers. To demonstrate accuracy and to assist other researchers, peer-reviewed articles always cite their sources in the form of footnotes, endnotes, and/or bibliographies. When an article is submitted to a peer-reviewed journal, the journal's editors send the article to other scholars in the same research field, that is, the author's peers. The peers look at the quality of the scholarship, its relevance to the field, and its appropriateness for the journal. To ensure integrity in the process, the reviewers' identities may be kept secret from the author, and the author's identity may be kept secret from the reviewers. Based on the reviewer's feedback, the article may be accepted pending suggested revisions, or rejected. You can't always tell a peer-reviewed journal by its cover. To determine whether or not a journal uses the peer review process, check the journal's website, Ulrich's periodicals directory online, or ask a librarian.
All right, so it kind of went over in general, like a short peer review process. There we go. You don't always know who's reviewing it, and you trust that the article is going to find people in their field. So whenever I've submitted an article for publication, I check kind of what fields I'm in, and they base it off of that. So they're looking at other people that have published similar work to do the review. Right now, I am doing a review for a textbook on undergraduate research and how to establish undergraduate research in different fields. And so they found me based off of other articles that I've been involved in. But I don't know if that person knows that I'm one of the reviewers or not. And you could always have the opportunity sometimes when they ask you to review, sometimes it's somebody you know, right? Because you're working in that field and those fields get small. You tend to meet those people at conferences, you tend to be in the same circles, to go to the same things, publish similar work. And so you have to say if there's a conflict or not. All right. Let's go. Yep. The journal's impact factor. Have you guys ever heard the term impact factor? No? Okay.
No.
Yeah. It wasn't something I was aware of until grad school. Certain journals have a higher impact factor, and the impact factor essentially, they can do a 2 or 5 year impact factor, but it is how widely distributed is that journal's articles and how much are they cited? Because if they're cited, that work is being continued. And they keep going on in the work, and they found that that work was valuable, right? And so the impact factor allows for you to kind of judge a little bit the quality or innovativeness of the person's work. If it's extremely innovative, different things like that. It doesn't always work that way, but you're gonna see a lot of in grad school there was always pressure. Like if you can get a Science or Nature article published, you're golden, right? Because they have the highest impact factors. And so when people look at your resume and stuff and they see something from Science or see you published in Nature, that means that they draw automatic conclusions. The work was innovative. It was at the forefront of the research, different things like that. And so different journals have different impact factors. I just pulled up a couple of different impact factors. Some of the journals, only about 1.9% have an impact factor greater than 10. Most have something lower than that. Science journal article has an 2-year impact factor of 56, and then has a 5-year impact factor of 54. As you go down, the impact factor decreases. When you get to the Journal of Medicinal Chemistry, I had to look at that. The impact factor is about 7.3. So even all of these journals are kind of in that top end of, or the top, I would say 4% of different journals for as far as impact factor.
And does that just depend on the quality of work, or work your way up the ladder kind of deal?
It depends on the quality of the work, and kind of the nature of the work. Is it on the forefront of something that needs to happen right away? Like, or how big is it? Like when the human genome was published, cover of Nature, right? They had the entire sequence for the entire human genome. Same thing happened when they did the mouse genome, right? It was new, it was comprehensive, it was complete, and it was the first time that was being done, right? It also involved, I don't know how many authors are on that actual original article, but it was a lot, right? It had a lot of different people. So if your work is innovative and kind of and timely as far as what people are interested in, right? You're going to have a higher chance of getting into a journal with a higher impact factor.
So when the impact factor refers to how valuable being published in that journal is, and then the 2 and 4 year is like within 2 years of publishing in that journal, you can capitalize upon it, more than 4 years out. Is that what it's saying?
No, so the 2-year, 4-year, 5-year impact factor, it looks at the journal itself and what articles were, see if I can go back here. The number of times published, okay, items published. Oh, I gotta stop touching my screen. The number of times it's published, right? Calculated by the number of times a journal is cited in the year by the number of articles published in the journal for the previous 2 to 5 years. So essentially, by citing the article, people are finding that article valuable and adding and building onto that work. And so the impact is more for, does it have an impact on overall science? Does it have an impact on potentially political or social areas where you have to change laws, policies? Does it have a policy factor to it? How that information, that journal is used. Does that make sense? Kind of.
Yeah, that was really helpful.
Good. All right. Um, so in general, peer review does a lot of this work for you, but when you are looking at information, just general scientific literacy, I see articles that come on my feed about different different case studies or different things that are happening. And if it's general news, I tend to go to the original article. But you can kind of evaluate some of this, some of your sources. I would not use a Newsweek article for your research, right? It's not providing any valuable information. It's providing commentary of what that author thinks is going on, which can be helpful in kind of adjusting your brain, especially if it's a scientific writer that's evaluating it. But in general, you want to keep kind of this in mind. Currency, I always get asked, how far back can I go for looking at different articles? It kind of depends on what you're looking at, right? If it's a disease that isn't studied very often, there might not be a lot of current articles in the last year, right? Okay, or even the last 5 years. So you might have to go back if you're revamping some and you're taking a new take on studying the disease. So currently, when was the information published or revised? It kind of depends on what you're doing. If you're doing work that is current, you know, keep it within the last couple of years. You can always reference something that historical, like in my thesis, I was looking at kind of proteins within the lipid bilayer. And so I actually cited in my thesis the original 1972 Singer fluid mosaic model, right? So did I do any work from that? No, but that was kind of the original source of the data and kind of the original idea of what I was looking at. It might not be in a short paper, but you know, a 250-page thesis, you can add whatever you want. So look at what you're doing. Relevance, you know, is it important? Is it on your topic? Who's the authority? Sometimes it's not easy to tell who the authority is, and sometimes you're not sure where the funding is. Most of those high impact journals, peer-reviewed journals, they all have to go through looking at if there's any conflict of interest. Where's your funding source, right? Are you getting funding? I always give the example of there was an oil spill. Oh God, I'm dating myself. Probably 20 years ago in the Gulf of Mexico near Louisiana. And if you saw a report that said everything went back to normal, you know, after 5 years, if it was from the University of Louisiana, that would be one thing. If it's from funding from BP oil, that's another thing, right? You can't always trust the motives in some of that. So hopefully the funding source or where the information coming is a neutral party. Right. Or in some cases, some researchers have patents, right? They have a patent for a technology, for a software program, for things like that. You might want to be a little leery of how they say it's the best thing ever and it should replace whatever system that is already in place, because they're going to make money off of it, right? I read an article, and this was a regular news article, but they traced back some of those misinformation campaigns to 12 different sources for the COVID vaccine. And it was the majority, it was like 90% of that was from 12 different people. And some of them were selling supplements, you know, as far as vitamins and different types of what I refer to as snake oils. And then others had different motives attached to that, right? Whether that be financial or personal. There are individuals that just are anti-vax. Okay. And whatever reason that they are, you know, they might want to promote their beliefs. Okay. So it's not always straightforward, but in general, peer review articles tend to be reliable. All right. Another little video.
If you want to succeed at your assignments or research, you should only use quality information, whether it's found in books, journal articles, or websites. Use the following criteria, also known as the CRAP test, to critically evaluate any source of information. Currency. When was the information published, updated, and/or revised? Is the information out of date for the topic? Relevance, or intended audience. How much information is presented? Does it provide a superficial treatment or a detailed analysis? Is the information related and relevant to your topic? Is the readership level appropriate, neither too simple nor too sophisticated? Authority. Not all books or journal articles in an academic library are scholarly. Who are the authors and/or editors, and what are their credentials? For journals, are articles peer-reviewed? That is, do they have the approval of other experts in the field? For books, are they published by scholarly presses, popular presses, or self-published? Accuracy and verifiability. Does the source match your understanding of the topic? Can you verify the claims in other sources? Never rely on just one source. Is there a bibliography or list of works cited? What types of sources and how many relevant sources are cited? This is an indication of the depth of the author's knowledge. Purpose and objectivity. Is the purpose stated? Is the subject approached from an objective standpoint? If not, what is the author's bias and how might it influence the information presented? Be wary, there may be more than one perspective on any given issue. Using these criteria, currency, relevance, authority, accuracy, and purpose, to critically evaluate sources of information will help ensure that you're only using quality sources of information. If you need more help with evaluating sources, visit one of our research help desks.
All right. So the actionable takeaways from this is identify the question or topic you are exploring. All right. Check for potential confirmation bias as you gather kind of a range of potential sources. Check for currency. When was it published? Whether it's been updated. Some articles are retracted, and I'll talk a little bit about retraction. Does your investigation require more recent information, or is some of that older information the only information that you can get? Some of those rare diseases that aren't talked about or studied as much. Are they relevant? Who's the intended audience? A source's authority, are they published by scientists or are they published by something else? Accuracy, what references have been used? Sometimes I find when I'm doing database searches and I find an article that I really, really like, even if it's a review article, I will go in, and like there's a piece of information and then there'll be the reference attached to it. And it's a lot easier now where you can just click on that. Then I can go look at that reference and see, if they interpreted it correctly or if I can use that paper in my work too, right? So it's a good way to find other, and then check for purpose. Is it meant to inform, persuade, entertain, or sell? Is there political, ideological, religious, or other types of frameworks involved? All right, retracted articles. Articles may be retracted when their findings are no longer considered trustworthy due to scientific misconduct, error, plagiarized, previous published work or they're found to violate ethical guidelines. Now, retractions sometimes take a while. So it's possible that you could use an article now that isn't retracted that may be retracted later, because we don't always have all of that information. Retractions can be full or partial. A partial retraction, there's only a portion of the article. That means like one of the figures of the work was flagged. There was an error in it. Usually there's a statement about why it was retracted and what the authors have done to resolve that. Or full retraction, which means the entire work was flagged. It could be the experimental design. It could be lots of different things. All right, so let's give an example. All right, and I know I'm talking more about vaccines, and my daughter, who's 19, is like, I don't know if they're going to want to talk about it. And I'm like, well, it's kind of my area. And so whatever your personal views, you know, we're talking about facts here. All right. Most of us have heard through social media, different sources, that the idea that the MMR vaccine was linked to autism, correct? So you've heard vaccines and autism. You've heard those two words probably together at some point. Most of that started with one article. It was done by Andrew Wakefield. And in this case, they later had to retract it. It took 12 years to retract. But on the surface, it looks like a general paper, right? It has several authors, which means several scientists had taken a look at it. Some of these authors, and if you're not familiar, like the primary author is the person who did most of the work, who did most of the writing. All of the other authors contributed in some way. They might have just read the paper and add results. They might have had a figure or two. They might have been involved in sample collection. So these were all people that were involved with that work. And, you know, you guys have the potential to be on a paper. You know, if you can get one good figure out of your research that gets published, your name goes on the paper, you know. So you get one good figure for your paper, and that'll be a nice kind of add to your resume. All right. So later on, after this came out, essentially what they did is they took 12 children between 6 mean age of 6, but range from 3 to 10. 11 of the 12 were boys, and they had a pre-existing history of pediatric. They were taken from the pediatric gastroenterology unit, and so they had several different types of underlying conditions. Okay. This paper, before it got retracted, got into kind of the mainstream media that MMR was linked to vaccines. Now, why it was retracted? One, it was a small case series. They only had 12 children. There was no controls. So there was no control population. It linked 3 common elements instead of looking at 2. Right? So they were looking at some of that gastrointestinal disease, nonspecific colitis, and pervasive developmental disorder, which is the autism factor into it. Okay. And both, mainly it was relied on, or the results were relied on parent recall and beliefs. Did their children act differently, or did something happen differently after they received the vaccine? No, there was no saying that this wasn't part of their pathology of their they already had something going on. How did that affect? Or there was no like baseline of how the children were. So this was published because it was a result, right? We published the result. there wasn't anything to that. And then they had a decade of vaccine studies with thousands and thousands and thousands of samples and with controls, and there was no link based off of that. So there was hundreds of vaccine studies and thousands of samples collected, and they found no link between autism and MMR vaccine. There were also some financial incentives for Andrew Wakefield. He had some ethical violations on his results, so that would be a personal bias, right? Because you get a financial incentive depending on what you were working on. And I don't remember all of the details of that. But finally, after 12 years, the entire paper was retracted. You can still find it. It's still at the Lancet Journal, but it has this big retracted red mark on it. So any papers that have been retracted should have a pretty wide retracted mark on them. And so you have to take them with a grain of salt and reading them, knowing that there are flaws in them. All right. However, even though it's retracted, it is still the number 4 most cited retracted paper. It keeps getting cited over and over and over. And most of that is not scientific, but it keeps getting cited. In general, the gold standard is peer review. There's nothing you can do about a paper that has been published, and if you use it before it was retracted, sometimes you can notice those different things, sometimes you can't. But you take every paper as kind of with a healthy sense of skepticism. Like, what were they doing here? Why are they doing this? I know that in general, we tend to just trust experts. In grad school, you are trained to be analytical of every paper and dissect every paper. You want to know exactly what's going on, why they use this, why didn't they use this technique instead. In general, the gold standard, though, is peer review, right? You can't be an expert on every field. You can't be an expert on every paper. So using peer-reviewed journals will help you get through kind of making sure that your source is reliable. All right. I have a small break built in for coffee. And you guys can use the bathroom if you need to. Let's see. I'm gonna pause. There was a question in the chat, and I just saw it during our break. About what do they mean by expression? And I assume that they're talking about how the DNA was expressed in the nucleus. So this kind of gets into how it depends on how they did the experiment. But they're looking for, essentially it's like PCR. They're looking for those fragments. And so they're looking for a fragment that's specific to the mRNA vaccine. And so it was a specific fragment of DNA that they were looking for. And when they do PCR, so there are a lot of different ways to do this. You could visualize it. So you could you could tag your gene that you want with like a fluorescent and then look to see where it visualizes, if it's in the nucleus or not. The other is you could separate the cell based off of parts. So basically you smash up the cell and you can do a sediment gradient and it divides out, where the mitochondria are, where the nucleus is, where the cytoplasm stuff is. And then you can look for if you separate those out, essentially, and then you put them into different test tubes. So this is the nuclear component, this is the cytoplasm component. And then you could do PCR on that fragment and see if the genes are expressed there. Does that that answer your question?
Do they do that based on density in a centrifuge?
Yes. So I've used sucrose gradients to kind of separate plasma membrane components and different parts of the cell. And in the sucrose gradient, you have like a certain percentage of sucrose, and then you spin it in a centrifuge, and all of the parts just kind of separate out into like a sedimentation gradient. And then based off of what gradient you used and your previous knowledge, you can kind of separate out where the nucleus is, or nuclear components would be. Yeah. And then what did you understand fully what it meant to have your name on a paper at the time that I published? I published my first paper in grad or as a postdoc. And by that time I knew what was going on. I didn't fully realize what the first authorship meant until I started getting contacted about my work. So who's ever labeled as first author is the primary contact for the paper because they did the majority of the work. And so, my last email was like 6 months ago from a paper I published 4 years ago, asking about what I did and how to approach different things. It wasn't until grad school that I learned kind of the dynamics of the authorship and what happened. My PI, or primary investigator, so every lab has a primary investigator. That's the lead person who's kind of directing the research. You guys are going to work with your PI as your research mentor, right? They kind of direct what the lab is doing for research, but they're also responsible for all the funding, for paying you, for paying for all the supplies. So there's a lot of stress that goes along with that if you want to be a primary investigator in an R1. If you're going to get funding from grants, there are requirements that you need to fulfill and to make sure that you're using that money wisely and that you're getting the results that you said that you were going to get. So that's usually the last person on a paper is the person who's doing most of the funding and directing the research, and then the first author. And then after that, you can be secondary author, like if you contribute one of the figures, but don't do the majority of the writing or the majority of the work. You can be a consultant sometimes. So in my grad school lab, we worked with clinical trials on basically it was a renal cancer, but they were looking at retraining our immune system and using these beads, these cell-sized beads coated with tumor proteins to see if that could induce the immune system to attack the cancer rather than using chemotherapy or something like that. It was supposed to be like a smart target, but we had clinicians who worked with the patients because we were PhDs. We needed an MD, so they were also on there because they were working directly with the patients and gathering samples. Yeah, I think that as an undergrad, I didn't grasp what authorship meant. Authorship is certainly important for depending on what you want to do. If you want to continue in the research field and get grants and continue on that route, authorship is extremely important because it shows proof that you are doing this work and you're doing it of a high quality, and then you can continue to get funding. And there's a little bit of status associated with it just in the scientific community, but the real thing is the kind of the funding. As a instructor at a community college, I don't have any pressure to publish. And this kind of goes into, we're a little bit off topic, but this kind of goes into kind of how do you want your life to look like, right? I watched my PI at an R1 institution. He was stressed all the time, right? All of the PIs, all of the head researchers, they just look stressed and exhausted, right? Didn't necessarily want that life. I wanted to be involved in science, and so teaching at a primary teaching institution versus an R1. So Augsburg, St. Olaf, community colleges, they typically don't have the pressure that you would see at an R1 institution. Yeah. So in the end, it didn't matter necessarily if I published in grad school or postdocs, but at that time I didn't know what I wanted to do either. Yeah. Someday I'll talk to you about my transition into grad school and how my application got dropped off the day before. I only applied to one grad school because I didn't know what I was going to do with my life. And my advisor, my mentor at college, said, you should go to grad school. I said, okay. And he happened to be going up to the school, so he dropped off my application for me. Yeah. But I would not recommend that for people. I would recommend applying to more than one and trying to get your life together a little bit faster. But, um, you know, we all have different paths and somehow it all worked out. All right. Any other questions on publishing, authorship, gene expression? No? Okay. All right. And if you guys notice stuff in the chat, just let me know. All right. And I am pretty sure I started recording again. Yeah. Okay. Oh, hi, Liz.
Hi, I just snuck on.
Oh, just in the moment where I was talking about my non-traditional grad school application.
Yes, I have to admit that stressed me out just a little bit. And I know it has like it's only just hearing it secondhand, but I'm like, oh my gosh.
Yeah. I hadn't taken the GRE yet, so that was contingent on my application. But yeah.
Wow. That's so cool though that you had an advisor that was willing to like take on that stress for you as well though and like drop your application off. Like that's we should all be lucky to have mentors like that.
Yeah.
Honestly.
Yes. All right. Well, we're just about to talk about databases.
Exciting. I'm just gonna hang out.
Okay. All right. So we talked about peer review. We talked about different articles, making sure to know if something's credible or not. Now we're going to get a little bit more into the research process. So when I look at the research process, I look at, the first is brainstorm. What are you looking at? Kind of have an idea in your head of key terms, different things that you want to look at. And then you learn and design, and then you can build. And so we're kind of going into that learn and design. We need to learn a little bit more. So there are different databases that you can use depending on your field. I tend to, just because I was in the biomedical field, I tend to use PubMed and the National Institute of Health database. Google Scholar is kind of general across all disciplines. It does contain both academic and kind of articles that aren't formally published. So not everything may be peer reviewed. And I'm going to link, or I've already uploaded this presentation to your Google folder. So any of these tutorials at the bottom, you can click on to make sure that you want to watch. I'll go over Google Scholar, but I have these tutorials for every single database that I'll look at today. And so you can kind of go depending on what your discipline is. Google Scholar covers anywhere from 2 to 100 million articles, estimated right now at about 389 million different articles that you can read. You can get abstracts, related articles, references, cited by, full text, and different export formats. So one thing I want to note, and I think I have it on my slide for Thursday, yeah, is that as you go and you're making notes, right, you want to make sure that you get the citation. So as you make a little note of like, oh, that's an interesting finding, and you put the finding down, make sure you put the citation right there with it. And however note you want to note that, that's fine. Most of these have links to the citation in whatever format that you want it in. And maybe Liz can correct me, but I don't necessarily use APA, MLA. I use whatever the journal wants me to use, but as long as I'm consistent in whatever I'm doing. So if I'm going to start with APA formatting, I'm going to stick with that for my presentation, right? I'm going to have it consistent. If I'm going to use MLA, that's fine too. I'm just going to be consistent, knowing that if I ever go to publish, each journal has its own requirement, and some don't use APA or MLA. They use their own type. I think the American Microbiology Association does that, so that's just AMA. Liz, you have a question?
Just a comment. I think that's great. One thing I would add is what I've done in the past would be to just go to the website of the journal the article is published, and you can find their like citation style noted on their website. So it could vary from discipline to discipline how they want to cite, in that field. So yeah, I think also too, Jessica, what you said was really helpful. Just being consistent is the most important thing.
Yeah. And this may be a question for your research mentor too. As you go with your research mentor, just say, is there a format that's preferred in this field? And, it'd be a simple question. And then you just consistently use that for what you need. All right. I do have a video. There we go. All right.
What's up, smart students? Welcome back to my channel.
All right. So this one particularly is using Google Scholar, but making sure that you can search for peer reviewed. This allows you to get the accuracy for peer reviewed articles and making sure that it's a quality article.
My name is Chelsea Seaburn, and today I want to talk about how you can find peer-reviewed articles in Google Scholar. Now, let's talk about Google Scholar and peer-reviewed articles before we jump into my computer and I give you the goods. So first things first, what is Google Scholar? Why is Google Scholar important to you as a college student? Well, simply put, Google Scholar, much like its big brother Google, which houses all the world's information, Google Scholar focuses on the world's academic information. It's a search engine. Just like Google, Google Scholar is a search engine housing various sources for academic purposes. So what does that mean? Well, if you go on Google Scholar, you can find things like journal articles, both published and unpublished. You can find an academic book, maybe even a conference piece or a court opinion piece. You can find dissertation papers, thesis papers, and of course, those peer-reviewed articles. Now, why does a peer review article matter to you as well? Simply put, peer-reviewed, which you might also hear called refereed, refers to the steps that this researcher took to publish that article. So I like to say that peer-reviewed articles are articles written by experts that are then reviewed by experts. You see, when an article is peer reviewed, that means that before it was published, other experts in that same given field reviewed that piece of work to determine its accuracy, the validity and quality of the research, and to make sure that reasonable conclusions were drawn. This is why peer-reviewed articles are important to college students, because they are going to be considered the most credible of all credible sources. And because I know that many of you turn to Google Scholar for research and finding credible sources, let me teach you 3 workarounds to weed out all of the sources so you can specifically find those peer-reviewed articles. All right, let's jump into my computer and go. And here we are. Welcome to your personal tutorial. So I have 3 different methods to show you on Google Scholar, and I have them listed from the first thing you should do to your last resort. So let's go ahead and start with a basic search because I want to show you the difference. So let's say your paper is about time management in college students. This is a basic search, having done nothing specific yet to Google Scholar, and we have all of these options here. Now, reading through the titles, let's say I'm interested in this 4th option down. So I click on it and I see that, okay, I have a preview of this article, but I don't have access to the full document. Okay, let's start there. Now, the first tip that I recommend doing for finding those peer-reviewed articles is to link your university's library to Google Scholar. And so what you're going to do is this drop-down menu on the left-hand side of the Google Scholar logo. You want to click that, go into your settings, and then click on this right here that says library links. It's going to bring you to a page that looks like this. And simply put, you're going to search for the university you're attending here to see if it is coordinated with Google Scholar. So let's say I went to Harvard because I was super, super smart. I'm going to type that in, search, and here we go. Harvard University Try Harvard Library. That's the name of Harvard University's academic online library. That was a mouthful. So I'm going to click that, select save, and then now look at the right-hand side where there's these PDF options. There is also the Try Harvard Library. This is telling me that these articles can be accessed through my university library. So going back to that 4th article down, I'm going to select my university's option, which will bring you, 1 second, to your university login page. Now, I can't log in, so you're going to have to follow this all the way through on your own. But the reason this is helpful is because in your university, you can filter out for peer-reviewed articles. You can search specifically for them. And so before you do anything else, you want to link your university to Google Scholar. That's method number 1. Let's move on to method number 2. So let's go back home so we can start over brand new. The second method I want to teach you how to do is to utilize Google Scholar's advanced search feature. And so it's not a perfect search for peer-reviewed articles, but it's pretty darn close. So again, let's go over to the drop-down menu. But this time, instead of going into settings, we're going to select advanced search. Here, we have a few different filter criterias in which we can return based on the data we put into these spaces. And so find articles with all of the words, with the exact phrase, with at least one of the words, without the words, and so on. I want you to pay attention to with the exact phrase. And in there, you're going to type in peer-reviewed. I suggest leaving out article because not all peer-reviewed will say article at the end of peer-reviewed articles, but now we're telling Google Scholar that we want to search for sources that have the phrase peer-reviewed. Once I've typed peer-reviewed in the exact phrase section, I want to think about which keywords that I want to associate with peer-reviewed. Let's say I'm doing the same search I was before, time management and college students. Excellent. Now I'm going to select search, and Google Scholar is going to return articles that have the keywords time management college students, and it's going to look for the exact phrase peer reviewed. So as you can see here, we have time management, college students, peer reviewed academic journals. Going down the line, she has published several peer reviewed academic papers, peer reviewed journal. Here we have peer reviewed. And so as you can see, now we're starting to pull up closer to what we want to find. But I do have to disclaim that anytime you find an article with this method, you want to make sure that peer reviewed, they are referring to the actual article you're trying to use. So let's click on this first one. And so basically what I'm telling you to do is that you just want to inquire and make sure that peer reviewed does pop up in reference to the article. As we can see, it does right there. So we're good. And it's not a phrase where they're describing that this author has written peer reviewed articles, but not this one. That's rare, but you just want to make sure to do the little step of looking for peer reviewed, making sure that it is indeed in the article. But this is going to get you to pretty close pulling back all peer-reviewed sources in Google Scholar. Now, I do want to point out that if you didn't want to use the advanced search feature, you could use the Boolean operator exact phrase, as you can see it populated here for us. And so I want to show you both ways, so I'm not sure how skilled you are with Boolean operators, but essentially, it's how you talk to Google Scholar with different symbols associated with the keywords. That's how you'll tell Google Scholar what you're looking for. And the double quotation surrounding a keyword, that tells Google Scholar that you're looking for an exact phrase match. So that's another way you could type this in. Now, the final way that I want to show you to find peer-reviewed articles on Google Scholar, it's not a shortcut, but it is a last resort. Keep it in your pocket. And that is to search the name of the journal you find the article in to see if that is a journal that publishes peer-reviewed articles. And so let's say this is the first example. You pull up this article, you don't see peer-reviewed anywhere within it, but you really want to use the information that's in it, but you need it to be peer-reviewed. So what you're going to do is use the name of the journal, which is Journal of Education Psychology. Perhaps you've never heard of this before. Go ahead and go to traditional Google and type that in to locate the original source that this came from. My little pro tip is to go ahead and use the exact phrase match Boolean operator here as well. So that way your searches are even more specific. And so now your job is just to inquire to see if this is a peer-reviewed journal. And right here, before even clicking on anything, we can see that it is, that the Journal of Education Psychology is a peer-reviewed academic journal. Great. Let's look at another example. Let's say, here's another article. We really want to use the information in it. Nowhere on here does it say peer-reviewed. So what are you going to do? You're going to search the journal, which is, in this case, Science Direct. So let's go back to Google and let's use our exact phrase match for Science Direct. It's going to bring me right here to the journal where that article is housed. And look at that, peer-reviewed journal articles. So we've qualified both of those articles. We're good to go. Now I want to show you an example of an instance where it may not be peer-reviewed. So once again, we really like this article. We see that it was published in ResearchGate. We're not sure exactly what ResearchGate is. So let's go back to Google, or you can quite literally type in ResearchGate there, but I'm going to use Google. Pardon me. Here we go. All right. So here we go. ResearchGate. We click on this and we see that this is a site all about research. It says discover research, access over 135 million publication pages. Well, that kind of sounds like you'd find peer-reviewed sources here, but nothing on here actually says peer-reviewed. So what I recommend doing is go back to Google, ResearchGate, and type in something such as, is this a peer-reviewed, even peer-reviewed journal? Simple as that. Ask Google the question, is ResearchGate a peer-reviewed journal? And right away, these are not peer-reviewed by ResearchGate. So what does that mean? It's that researchers' Facebook. So ResearchGate is a social media for research. It's a sort of researchers' Facebook with an option to upload papers or reprints of papers that were published elsewhere. So basically, what that tells you is that you might find a peer-reviewed paper on ResearchGate, but ResearchGate itself is not enough to say that it's peer-reviewed because there's a handful of different types of research papers coming in. Like I said, this is the last resort, but it's important you know how to search to figure out if they are peer-reviewed or not, if that's what you need for your research papers. And we're back. All right, friends, I hope you found.
All right. So I think that a lot of that was good information. In the end, ResearchGate is like a database. So it's not a journal in itself. So be careful with that, because I noticed like there was like if there was a name of the journal below, it was just found on ResearchGate, right? And so you could look at the name of the journal itself to find out if it was peer reviewed. ResearchGate itself doesn't control what comes in and what's published on that site. Cool? All right,
On that tutorial on.
there we go. All right, this is the one I'm most familiar with. PubMed is for biomedical research. Contains right now over 37 million articles. It has the abstracts, it has related articles, it has reference, it includes cited by. I'm gonna pull up, I should have just linked it right to. I'm just gonna pull up one of my sites. Everybody like sees all my tabs open and they wonder if my computer's gonna explode. Yeah, it might. I melted the hard drive on my last computer, so. All right. So PubMed is the National Library of Medicine. In general, if you want to go to the main page, you know, you can search. We can. I think before I just searched congenital heart disease. It'll come up with a lot of different. Some of them you can have the free article. You can essentially filter the results for anything with full free text. If you want the article itself, your library might have different access. So I know I had to actually ask Liz to get access to the biomedical library at the U of M because I wanted a particular article. So your home institution may have different things. Otherwise, you can always do an interlibrary loan. So if you go through your home institution and ask the library to send you that article, most of the time you can get it within 24 to 48 hours and they will send you the article. There's a lot of crossover and coordination between libraries to help you get access to what you need to for academic journals. All right. One thing that I do like about the PubMed is that I can get the citation right there. I can do it in whatever format that I want. I can be consistent. If I want the APA format, right, I can just copy it and put it into it. You can also download it into a different type of software that does bibliographies. So as you're going, I can't stress this enough because I have been in that position where I've done research, I'm writing the paper, I'm like, oh my God, where did I see that? Where was that? And then I waste another 3 hours of my time trying to find it because I'm not in the same rabbit hole that I was before when I originally started finding the paper. And so not always knowing exactly what you're looking for. So I encourage you when you're starting to take notes and write notes, start keeping that list of citations together and like little notes about that article that, you know, the main findings that you found were interesting. Also, if I'm really interested in a particular researcher's work, you know, I can go and now it is all of the articles that they've ever been an authorship on. Right? I can limit the article type. I can, limit the publication date to make sure that it's current. So you can do lots of different things. Almost all of them will have an abstract available. So, and I will talk about it a little bit more when we're reading literature, and I'm not sure if it's on this slide or not, about when you're going through and like when I typed in congenital heart disease, I'm not going to read 200,000 different articles, right? You have to be picky. Is this relevant to what you're looking at? And the abstract should be able to tell you that. All right. Now we'll go back to our presentation. All right, and then I have a tutorial for that. For computer science, I asked some experts in that. A lot of them use the ACM digital library, contains over, almost 3 million articles. It has a lot of the same features. I'm not as familiar with that, but I do have the tutorial available for you guys to kind of look at that. Another computer science, electrical engineering, and electronic database includes the IEEE Explorer Digital Library. Again, I mean, this one has almost 5 million articles, and it's based more on articles related to engineering and computer science. Math. So if you're a math person, math major, and you want to know what the latest academic math research is, this is published and created by the American Mathematical Society, contains almost 4 million articles, and you can, you can go ahead and use that, kind of going all over the place. And then there's the American Society of Civil Engineers. So depending on your field, you may use this in the civil engineering. Again, it contains proceedings, books, standards, manuals, magazines, similar to what you'd find on Google Scholar, but it's just more geared towards math. All right, some key takeaways for avoid typing the whole research question. You want to use keywords and use several keywords. So I put in congenital heart disease into my PubMed search, but it's 200,000 articles. So what do I also want to look at? Do I want to look at a specific disease, like hypoplastic left heart syndrome? So I put that in there, and that'll limit that as well. So try to use keywords. Be willing to use a synonym. I'm trying to think of an example of, so instead of heart, maybe I use cardiac, right? Look at what words mean similar things, especially early on when you're studying something, because different fields may refer to the same thing, just slightly different. And you want to make sure that you capture what you need to. And then use and or to retrieve more results or to limit your results. All right. And then I have time for another coffee break. I'm trying to look at what else we have here. We have about half an hour. Do you guys want to push through, or you want to take a little break?
I could keep going.
Up to you guys.
All right. Well, if you have to, you guys are adults, so do what you need to do. All right. Let's talk about reading the scientific paper. I'm going to, at the end of this day, I'm going to assign you some scientific papers and you're going to present them tomorrow. I know that's a tight turnaround. We're not expecting perfection, right? So, first, let's look at the anatomy of a scientific paper. There's a title, you have the authors, you have an abstract, an introduction, methods, results, discussion, and references. All right. So in the abstract, this gives you the brief overview. the majority of your reading starting out is probably going to be the abstracts. You want to sort out, is this relevant to the question that I'm asking? Their abstract explains why the authors did the experiment, how they did it, and what they found out. So a lot of times from the abstract, you should be able to tell if this article is worth reading. The introduction explains, why did they bother doing this experiment? What's the background? What led them to get to where they are now? Why was this a problem initially? Why were they interested in it? So it explains the prior research, what the accepted understanding is in the field. All right. The methods explains kind of, it has to be explained. It explains what the researchers did to get the results and how they set up the experiment. And it has to be detailed enough for another person to replicate that experiment. That's the general standard. You want to, when you write your results, you want to make sure that anybody else could then do the same thing for that experiment. What I recommend early on in reading is to kind of skip this section, right? If it becomes part of your paper, you're definitely want to read it. You may not fully understand it. I don't think I understood people explaining their PCR until I actually did PCR. I don't think I fully understood some of the cell microscopy stuff until I actually did the microscopy. And so some of this will come with experience and fully understanding it. But for what you are doing, especially early on, you can skip the materials and methods until you know that that's a paper that you want to use, right? And then you can work on understanding some of those materials and methods. The results, what the data is, the data the authors use to reach their conclusions. You want to, like, understanding the figures and tables is important in understanding the paper, right? And when we do journal club, explaining those tables and figures and how it happened. So if I was looking at journal club and I was going to explain this figure to you, I would have to know, where did we find the red apples? What are we looking at? Because right now all I see is red, yellow, green, and then a table describing color, right? What are they doing here? I need to be able to tell whoever I'm going to present to exactly what they did and why they did it. So make sure you understand the results section. And then the discussion or conclusion is where the author connects the dots, right? What does this data mean? What does this mean for the field? Why it supports their conclusion? Make sure that you are starting kind of that analytical brain. Remember, we started to question, is this valid or not? Why did they do it that way? That doesn't necessarily make sense. Keep that skepticism when you're reading these papers, right? Would you conclude the same findings? Why or why not? Right? That will help you become a kind of a critical reader of the papers. And if you're not sure, right? That's where you ask your research mentors, your instructors, your postdocs or other graduate students in the lab saying, I don't get this, and, and just kind of ask them. All right. And then list of references and bibliography. The only time I use this while I'm researching or skimming the kind of the databases is if I'm reading a paper and I see that one of their sources, like the previous work, showed that X caused Y or whatever, right? And it was part of a bigger paper, but I want to then look at that because it's relevant to exactly what I was trying to get to. Then I'm going to click on that and go to that paper as well. So the references are there to tell you where kind of some of that information. All right. Now we get to Journal Club. Anybody have questions about the anatomy of a paper and how it's structured? Most of you at some point have either looked at a scientific paper or had to do, even if it was in, I remember in middle school, I did a science project where I had 3 different colored bird feeders, right? And I watched which birds came to which an hour a day or whatever. But I had to have a title, an introduction, materials and methods. So we're somewhat familiar with that concept. But we don't always know why, and why, when Devin asked about the authorship, why is that important? How is that structured? We're learning more and more about the anatomy of the paper and how it's structured. All right. Any questions? Now that Liz is here, you don't have to be quiet.
No, all of that made a lot of sense to me.
Okay. All right. I'm just teasing Liz. All right. So let's talk about a journal club. I don't know if any of you participated in a journal club. It's similar to like different reading circles where if somebody reads, they all read the same book and then they get together and talk about it. Journal club is where you're reading a journal article and they're getting together. My first experience with journal club was in graduate school. I didn't exactly know what that meant. I was nervous about it. Luckily, my first time showing up, I didn't have to present right away, but essentially we had a department journal club where we assigned one person every week, that date. And that person was in charge of picking out an article, emailing it to everybody else so that they could read it beforehand. And sometimes the people that you're presenting for were very familiar with that article, and sometimes they kind of skimmed it over. So you're going to get a lot of just kind of various questions of whether they read the article or they didn't read, or sometimes it's very detailed because they know exactly what was going on in there. But then you're assigned a date, and so you email the article to everybody else. And when that day comes, you prepare a presentation that goes over the article. Like, why did they do this? Essentially, kind of, what the title was, why did they do this? And then you go over detailed, here was figure 1. This is the procedures that they use. This is what the information that they got. This was kind of the conclusions that they drew. And the first time you do it, especially in graduate school, where, you're the young student, not familiar with all this, and you're presenting to a group of, PhD, MD doctors that have been in this field for a while, it's very intimidating. All right. It's not meant to be intimidating. The idea of journal club is to kind of discuss the paper as a group and be analytical about it, critique the experimental design, critique the methods. Keep in mind, they are not critiquing you or your presentation skills. They are critiquing the paper, right? And you won't know everything about that paper. That's okay. You might not know all of the background, and you just have to say, I'm not sure, or I'm not sure, but I think it might be related to this if you have some idea, right? So the goal of the journal club is not to critique you or your presentation, it's to critique the paper. So try to, stay focused on the work itself. All right, let's talk a little bit about this.
Today, I'm going to talk about journal clubs, what it is, how do you prepare for it, and how do you present it. So what is a journal club? A journal club is a session where a group of people, clinicians, researchers, students sit together and discuss one journal article. So there are several goals to this. 1, learn how to critically analyze or appraise a journal article. Number 2, get to the forefront of evidence. If you are a clinician, you want to learn about evidence-based medicine. 3, be part of the scientific discussion. So preparation stage. The question I get all the time is, how do you choose an article, or what type of article? The first criteria is to choose a primary research article, not a review article. This is because you want to be able to discuss the methods, results, and discussion of the paper. And the review paper does not have any of these components. The second criteria is to choose something you're interested in and that you can learn something from. The third criteria, understand who your audience is. Clinicians, are they basic scientists, maybe they're mixed, and be sure to discuss with your supervisor to make sure that the article is appropriate for the audience. One of the big mistakes I made when I was a renal fellow was that I chose a mouse model study for a journal club, and I had no background knowledge about that. And my audience were mainly clinicians, and so we had a hard time understanding the paper, and so it wasn't a very meaningful discussion. All right, next step, read and critically analyze the paper. We also call this step critical appraisal. So what does it mean to critically appraise a paper? So you want to know 4 things. What is the aim of the study? Can you trust the method? Can you trust the results? And then what are the results? Finally, how can you use the results? So I'm going to give the overview here because I will be talking much deeper as we talk about the presentation stage. And as you're reading the paper, be sure to also read other articles that can supplement this research paper. For example, make sure you read the supplementary materials or look at the protocol paper that has been published about this paper. That's because oftentimes the details of the methods and results are either in the supplement section or in the protocol paper. I know conducting a research project is an overwhelming process, so I need the idea to paper blueprint for you. This blueprint takes you through a 7-step process from idea generation to paper submission. So be sure to get a copy by clicking the link below. Next, let's talk about the actual presentation. Typically, journal clubs are presented using PowerPoint slide. And before you even do the actual presentation, be sure to send out the journal article ahead of time so that the audience can read it. But also do understand that some may not read it. So your presentation needs to have enough information that people who have not read it could understand your presentation. As you present your journal club, make sure you go in this order. First, the introduction, then methods, results, and discussion. Don't mix them up. I've seen this mistake done many times, people jumping back and forth, sometimes intro, sometimes back into the results. Your audience are usually used to the same structure. So when you go back and forth, it's very confusing. So the first section, the introduction, make sure you talk about what the study aim is, what has been done before, and why is it important. The next one, the methods. Spend the most time here because this is where you determine whether you can trust the results or if you can use the results. Some questions you should be asking yourself. Does the study design answer the research question? Who is the study population? What's the inclusion and exclusion criteria? Are they appropriate? Any selection bias? Next, think about the definition. How do they define the outcome, the intervention, or the clinical condition? And then the outcomes. Are they clinically important? Or are they just surrogate outcomes? Then look at the data. What type of data did they collect? How did they collect the data? Any issues when they were collecting the data? Next, the statistical methods. Here, you will need basic understanding of different types of methods. Briefly, you need to make sure you describe the descriptive statistics and inferential statistics. You finally look at if they have used other ways to limit certain biases. Next, the results section. Here, make sure you start by talking about the sample. Describe the sample. Look at the demographic table. And when you are presenting, present findings from both the figures and tables, and also from the main body of the results section. So when you read the main body, it's really to get the interpretation of the results. As you're going through the tables, also make sure you look at the margins of error. That means look at the confidence intervals and the p-values. Are the results statistically significant, or are they clinically significant? Next, the discussion. How did the authors link their study findings with other studies? Do other studies support their finding or not? If not, why? What is the implication of the study? What's next? Then limitations. Here, think about limitations in terms of limitations inherent to the study, limitations regarding the study population, maybe limitations regarding the sample size. Also look into how the authors try to overcome their limitations. One extra way to add flavor to the discussion section is also to look at editorials or other papers that have cited this particular journal article. Finally, ask yourself, can I use it? What's the generalizability of this paper? Can the study finding be used to other population? Another question is applicability. That means, can the study result be used for my own patients? Final tips. Number one, make sure you spend enough time reading the paper and understanding the paper. I would read the paper at least 2 or 3 times, look at the results properly. And then in terms of presentation, especially when it comes to tables and figures, do not just copy and paste a whole table and put it into the presentation, because usually it becomes way too busy. So if there are certain parts you want to highlight, crop them and enlarge them. So there you go. This is how you prepare for a journal club presentation. If you like this video, please give me a thumbs up. I'll see you in the next video. Today, I'm going to talk about
There we go. All right. So that was some tips for journal club. Know the background material. I know that for tomorrow's journal club, you might not have time to do all of the background material, right? We're kind of fast tracking. This is supposed to be an introduction to kind of how things are done, and you know, if you're gonna make mistakes, do that with me, right? I'm a mentor, not a teacher, right? I don't need a final product. If it's not all final done, that's okay. Like, make sure your presentation is concise. So try to make sure that, you know, you pinpoint what exactly you want to say and don't try to extrapolate on that. Simplify unfamiliar concepts, especially in, you know, you're just starting out in a different research field. Some of these, and particularly our audience, is the rest of your students here, you know, the same people that are on this group. Make sure that you can summarize it in a way that they will understand. Ask yourself questions about the paper before you present, and ask specific questions to the members of the journal club. So when you're discussing, you can ask more questions. Really, it's meant to be conversational. I mean, you're presenting, but hopefully the audience will participate in your presentation. All right, now I get assignment for tomorrow. Tomorrow, I'm gonna have you read, you only have to present on one of the articles, but I'm gonna have you read these 5 articles. You don't necessarily have to apply the crap test, although you should just kind of do that in general when you're thinking about it. These are the articles that are done by the research mentors in this program. All right, so we have Dr. Keller-Ross, we have Dr. Yang, we have Dr. Huang, we have, oops, we're back. Maybe. There we go. Dr. McGovern. And then this was a supplemental. It was sent by Peter's group that he recommended for reading. And so I have 5 different articles for that. I also have some articles for the retracted articles. What I'd like to do is divide it up. So we definitely go over these 5 articles. And I want you all to kind of read these 5 articles, but when we present, we can talk about other articles as well. All right. In your Google folder, right, go back to the Google folder. On day 1, scientific literacy, I have already pulled all of these articles for you, journal club articles. These are the 5 articles that we have. And then I have retracted articles. I have that original Wakefield. And then basically these are the top 4 most cited retracted articles found on the web. So they still continue to be cited, even though they've been retracted. All right. So how many people do we have on our list here? I'm just trying to get a full view of all of the students here.
I do have a question about retracted articles.
Yeah,
Is there anything that tells or explains what was the issue with the retractions?
yes. So typically there is a link. I'm gonna have to, let's see. I'm not sure which link I had it on. I had to Google retracted. Um, 10 most top highly retracted articles. So if I click on that, it takes me to the article, and you can view the retraction, right? So it tells you there were irregularities in the randomization procedures. And so they have reanalyzed and have published the new report. And so you can actually go to the new report as well. So there will be reasons for the retraction. All right. So of the papers that we have, I can take volunteers and then I can start assigning people. The cardiovascular effects of transcranial direct stimulation. They have solute flow and particle and transport in aquatic ecosystems. Ugh. A salt house project designing for death, structural and functional connectivity and hydrocephalus, and an overview of the status and challenges of CO2 storage in minerals and geological formations. So there.
I can take Dr. Judy Yang.
Okay.
And then I won't be present tomorrow, but if you'd like, I can make a pre-recorded video and
That'd be
submit
great.
that to you before then.
Yeah, that'd be great. And then I can show the class tomorrow. Okay. Colton. Any other?
I could do the CO2 article.
Yeah. Okay.
I volunteer for the Salt House one.
Okay. Sometimes that was Ashley.
Yep.
Yep.
I can do the first one.
Sarah.
Yes.
All right. Anybody want to do Dr. McGovern's connectivity and hydrocephaly?
I could do that
Okay.
one.
All right. Make sure I spell that right. And then who wants to do some retracted articles?
I can do one of them.
Yeah, Chris. Okay. Do you have a preference on, let's see what we've got here? There, no. Forget where I had that Google folder now. See, this is the importance of, or not having that many tabs open and bookmarking. So we'll just do retracted articles again. I'm not sure why my husband decided to mow the lawn. So if you saw me moving from room to room, that was the case. All right. So I think I picked the top 4. Anyone do prevention of cardiovascular disease?
I can do that one.
Okay. And then that will be found when you go to the Google article, right? It'll be right in here in the retracted articles. Go back. Anybody want to do medication adherence measure? No. Micro RNA signatures and tumor-derived exosomes?
I'll do that one.
Okay. And the Wakefield paper? Or the visceral fat and insulin? Or does everybody have? I think
I'll do the Wakefield.
okay. All right. And then I think we only had one student missing today, so I will take care of that and assign her paper. But yeah. All right.
And so just really quick, we should read all 10, but be very familiar with R1.
Read the 5 that are related to the research that we're doing. And then if you have a retracted one, you're going to present on that, but make sure you read the other 5. Does that make sense? Just for
Yeah,
the sake of
perfect sense.
time, because I'm only leaving, I've taken up about 2.5 hours of your day already. And I want to be conscious of your time, and your time is valuable. So I want to make sure that you have enough time. Otherwise I would have you read probably more, but
Yeah, I think that sounds great.
okay. Any other so if you have any questions about presenting, try to have a PowerPoint. What I would suggest is let's plan on, around 15 minutes for a presentation. In a graduate school journal club, it goes an hour. So you have an hour to present that whole, but there's a lot more discussion. It goes into a lot more detail than maybe we need to right now. So this is kind of just a mini journal club. So plan on 15 minutes to present your article. And we'll do that tomorrow, starting at 9 a.m. And that's kind of what the agenda is for tomorrow. Other questions? You guys ready to be done with me for today?
I'm curious what Wednesday looks like in a little bit more detail, because I'm available from 9 to 10. So I'm excited to see and hear Amanda's presentation, but I also kind of want to know what I'm missing out on the next 2 hours, and is there anything I should do in light of that?
So, what I would make sure, I have a little bit of a talk prepared for from 10 to whatever, and I'll record that so that we can have that. It's on research elements, and honestly, I haven't fully got through what I want to all say, but basically the components of different types of research applicate, like whether it's applied or basic research. I'm still kind of working on some of this, types of scientific study. What else are we going to go over? Data literacy, scientific writing, what that looks like, kind of guides for effective scientific writing. That should be not as extensive as it was today. So hopefully it's, less than an hour, so that'll give you guys time. I'm also going based off of the schedule. And then it looks like on Thursday, I will talk a little bit about a research question. I'm not sure how I'm going to approach that yet, but we'll talk about kind of the components of that. So hopefully there'll be the research talk and then, me talking for hopefully less than an hour, right? I want to give you guys time and allow you to do things. So whatever time that it takes us to do through all the journal articles, if you've got extra time after that, make sure you definitely, everybody reads Amanda Keller Ross for the next day, right? And then after we talk about research elements, definitely make sure that you read Dr. Huang's paper. Does that make sense? So
Yeah, perfect sense. And then I'll be able to find that on the YouTube channel, correct?
yeah, so and I haven't told Liz this yet, but yeah, because rather than dealing with Kaltura
Yeah.
and logins, I'll put it to my YouTube channel and you should be able to see it from there.
Yeah, that's
So-
a great plan.
Okay.
I have
Other,
a quick, I have a
Yeah.
quick question. If we miss days, is it like, do we watch the video and then submit a reflection about it
Liz, how would you
for
like
our
to do
like
that?
meetings?
Yeah,
Yeah, I think I know that we haven't talked about what we are going to do if students miss, but I think in the case that there's a recording, I think asking for a reflection is appropriate. We just want to make sure that you're keeping up with the material, and that you're actually watching the material. So, I mean, I think, yeah, having a reflection would be good. Similar to the virtual EPI program, Ashley, and I can include instructions for this in our shared folder and let everybody know when they're available.
Okay, because that's what I was asking.
yeah.
Yeah, it's a
It would
great
be a
question.
little bit harder on like days that you have research, you're gonna have to work that out with your research mentor. And they might be able to say, if you can't come in that morning, come in the afternoon. They might be able to say, hey, why don't you just come in on Wednesday? So those days, just make sure you work out with your research mentor. Lab skills days, I can't really make up.
Yeah.
So, I mean, I can potentially find videos that cover that, but it's still not the same as actually performing the experiments.
Yeah, I got it.
Okay.
And in the matter of like record keeping, Liz, should we just have sort of a record of what hours, what days we missed, the reflections from watching those videos, just kind of all of that, maybe in like a spreadsheet or a folder for you?
Yeah, that's a great question. So I'm gonna do my best to like keep track of attendance on these virtual days. And I'm personally tracking that. So there's no need to track your own attendance per se. And then in terms of like for the in-person meetings, I'll check in with the instructors to see like if anybody was absent. In terms of any reflections, to make up, it could just be added to our shared Google folder. And you would just have a folder with your name on it, and you can upload any materials that you need to essentially turn in can go in your own folder. Does that make sense?
That makes perfect sense. And then, so like I know exactly what my schedule is. So would it be helpful to maybe have that in my folder under my name for you guys to reference? And obviously I'll reach out as well.
Absolutely. I think that's a great tool we can use. So if you have any schedules that you'd like to upload to a folder with your name on it, please go ahead and do so. And then that way we can plan ahead for any accommodations that we need to make.
Awesome.
Yeah, we're definitely, happy to make these accommodations. So I guess the sooner that we know, the better.
All right. And Liz, did you want a folder? I don't know if you guys can still see my screen.
Oh yeah, we can.
Okay. Did you want a folder added for assignments or turned in materials?
Yeah, we could do that. That could be like a master folder, and then inside that folder, everybody can add their own with their name.
There we go. That way we don't have multiple folders on this main page.
Yeah, that's
So
perfect.
just a quick question, like the video that I'll make tonight, should I put that in that folder under my name, under submitted materials, or would it be better for me to just email it directly to you, Dr. Bell?
It's up to you. You can submit it on there and then just send me a link so that if you send me a direct link, that would be great because then that'll make it easier for me to incorporate in our session tomorrow.
Perfect. Okay.
All right. Other questions?
I did have a question for the group. I know that you talked about this during orientation, but I just wanted to circle back. I know that it's the first week and all of our meetings are virtual, but for next week, there are some in-person meetings. And I know there was some questions about transportation. So I wanted to make sure, is anybody willing to carpool, or what's sort of our situation here with the in-person meetings and making sure that everybody has access and can get there?
So I live in the South Metro in Shakti. I don't know if anyone lives this way, but I will be driving to all the Monday, Wednesday in-person meetings. So I'm definitely available to carpool if anyone needs that.
I believe, Chris, you would be the only person that would need help with transportation. Is anybody else, is everybody else okay, or is it just Chris? And Chris, you're all moved into campus, is that right?
Not yet. I plan to move in like on Friday.
Okay, so Friday the 14th?
Uh, yes. Yeah.
Okay. So, Colton, Chris is living on the Twin Cities campus for the summer in Pioneer Hall. Is that still, I thank you as well for offering to help with the carpooling.
I'm not super familiar with U of M campus. My first time there was on Friday. But if that seems like a viable thing, then we could definitely have that arranged.
Yeah, and even if it was just meet or like I'll pick you up in front of this coffee shop or if it's something down below in front of the house.
Right.
Because the U of M is
Yeah.
pretty close to Augsburg. It's like a mile away from Augsburg. So some of those sites, and it's near the Minneapolis camp or college too. And you would have to go through Minneapolis to get to Century. So
Well, and that's exactly what I was gonna say. I think I
Yeah.
would take what, 63, so it's already. Yeah, definitely you can reach out to me. Do you have my phone number, Chris?
No, I do not.
Do you wanna jot that down?
I actually like don't have, I guess, a working phone number. So do you have like, it's
Oh,
just Gmail
this
through chat?
if we get permission from the students, can we put their names and phone contact information in the Google Drive?
Yeah, as long as we have like their, approval, I think that's okay.
Okay.
Okay. I will be transparent though. I feel like for that situation to work, I would need a phone number.
Okay. I probably can't provide that though, so.
Okay, understood.
Well, Chris, let me email you, and we can chat about transportation, okay?
Okay.
All right. Okay. I think that kind of covers anything that I needed.
All right. I could probably stop recording. I probably could have done that.